Hello, I'm Xiyu Peng. I'm a postdoc fellow in Memory Sloan Kettering Cancer Center. Today, I'm going to present a poster presentation, Accurate Estimation of Haplotypes and Abundances from Illumina Amplicon Data by AmpliCI. This is a joint work with my PhD advisor, Dr. DAUMI. Amplicon sequencing has been widely applied to explore genetic heterogeneity in populations and identify real mutations in populations, like microbial communities, viruses subpopulations, and cancer-driver genes. One main challenge is how to confidently distinguish real biological variants from noisy sequences. Here, I'm going to present an open-source software for clustering Amplicon sequences. Given a sample with errors from diverse populations of amplicons, AmpliCI provides high-resolution, reference-free, probabilistic model-based approaches to identify distinct biological sequences, as well as estimate their abundances or deduplicate abundances if UMIs are provided. Due to time limitation, I will just present the key ideas for clustering sequences with or without UMIs. We propose to apply the finite mixture model for clustering Amplicon sequences without UMIs. The mixture model assumes each read, along with a quality score sequence, is generated from K clusters. To calculate the sequence distance using the mixture model, we propose an error model. It considers both substitution and index errors in the transition probabilities, in which we use quality information for high resolution. In order to overcome the high computation load, we propose a greedy haplotype selection for fast inference on the mixture model for clustering. This is a t-SNP plot of 3,000 reads from 12 bacterial strains. At the beginning, we will select the most abundant unique sequence A as the haplotype. Then we try to measure the effect of haplotype A and subtract the effect of A from the observed dataset. That for each remaining unique sequence, we update abundance as times we observed minus estimate times misread from A. Then we select the next most abundant unique sequence B as haplotype. We use the same way to subtract the effect of B from the dataset. We continue to select haplotypes until abundances of all unique sequences are below a threshold value. Using this strategy, we can gradually build our haplotype set, and the algorithm could approximately maximize the likelihood function of the mixture model. As the result, we will find all 12 clusters for this dataset. For dataset containing sequences attached with UMIs, we propose another novel approach for clustering. We recommend to use UMIs here for several reasons, especially for dataset with high PCR duplication. With UMIs, we are able to estimate the deduplicate abundance for each biological sequence. For example, here I show the deduplicated abundances are 3, 2, 1 for the 3 distinct biological sequences. The underlying model is a one-step hidden Markov model which models both haplotypes and UMI sequences. This model considers both UMI errors and collisions and provides accurate estimates of the duplicate abundances of haplotypes, which is the sum of gamma SK for haplotype HK. Gamma is the transition probability between UMIs and haplotypes. Compared to other UMI-aware clustering tools, our proposed method has several advantages. It has high resolution since it takes into account both UMIs and quality information, and it can recognize UMI collisions, that the same UMI attaches to 2 distinct biological sequences. For better illustration, here I give an example for UMI collision we found in an HIV dataset. Only our method can accurately identify two distinct haplotypes attached with the same UMI. In the end, I want to mention that the AmpliCI v2 version now is available in the GitHub for clustering Amplicon sequences with or without UMIs. The core clustering algorithm now is available as a standalone C library for use in your own applications. It's reference-free, high-resolution, and big data scalable. If you are interested, please check out the papers for results and details. You can also find me through the Twitter and email. Thank you for listening.